fluorescence Western blot imaging, Western blot chemiluminescence imaging, tissue fluorescence imaging, fluorescent protein expression in cells or microorganisms, protein gel imaging, as well as nucleic acid gel imaging and gel excision. See how the Service Bio SCG W6000 can cover all 6 major experimental imaging scenarios with just one instrument. Let's start with fluorescence Western blot imaging. Equipped with 3 fluorescence channels, the Service Bio SCG W6000 enables multiplex analysis on the same blotting membrane. Fluorescent protein markers are costly. One costs twice as much as a standard pre-stained markers, and they are infrequently used. The exclusive inversion function of the SCG W6000 instantly displays all bands of pre-stained markers under dark field, acting as an ideal substitute for fluorescent markers to save time and lab funds. You can also choose cumulative exposure to enhance signals, or drag the time axis to adjust band intensity. After imaging, the source files can be analyzed using Service Bio Western blot grayscale analysis software. You can adjust the timeline to obtain different band intensities, and select individual channels for separate analysis. The software supports one-click composite imaging of multiplex fluorescent antibody signals from the same blot, and generates high-quality multiplex staining images even when primary antibodies are derived from the same host species. After fluorescence imaging, let's move on to the 4 most common issues in Western blot chemiluminescence and see how the SCG W6000 can help us get perfect results in a single run. For faint blot signals, cumulative exposure can be activated to accumulate light and strengthen band signals. For strong signals prone to overexposure within 1 second, exposure time can be finely tuned in 0.1 second increments to obtain optimal band. When the exposed marker bands interfere with the interpretation of your results, you can simply use the marker removal function to restore the true marker bands with one click. Conventional imaging systems can only process membranes one by one when multiple membranes are involved. This not only occupies the instrument for a long time, but also causes signal loss on the blots that are imaged later while they wait for exposure. The SCG W6000 supports simultaneous exposure of multiple blots. Captured composite files can be split into single blot files with one click for independent downstream analysis, greatly improving experimental throughput. All saved raw files remain compatible with the Western blot grayscale analysis software for post-capture timeline adjustment and flexible selection of band signals at any intensity level. The SCG W6000 makes Western blot results clear, publication-ready, and easy to obtain. Meanwhile, the Service Bio SCG W6000 can capture the autofluorescence of tissue samples, enabling real-time region of interest selection and quantitative analysis of tissue fluorescence intensity. Fluorescence-positive regions can be selectively sampled and sectioned according to tissue fluorescence distribution. Save the autofluorescence signal using the scanner to avoid signal loss during sectioning and experimental procedures. Then, the scanning results of autofluorescence and immunofluorescence can be merged to generate composite images combining tissue autofluorescence and multiplex fluorescent labeling. All autofluorescence datasets can also be analyzed via the Service Bio Western blot grayscale analysis software. In addition, when using cells or microorganisms to express fluorescent proteins, the SCG W6000 can also be used for imaging, allowing you to determine the expression level of fluorescent proteins based on fluorescence intensity. Having witnessed the excellent performance of the SCG W6000 in Western blot and tissue fluorescence imaging, let's move on to its gel imaging functions. First is protein gel imaging. Color or grayscale gel capture modes are optional, and color protein gels deliver crystal clear band visualization at a glance. The nucleic acid gel imaging function supports ultraviolet imaging with various common dyes, including ethidium bromide, cybergreen, and goldview. For gel excision, simply attach the UV protection shield and click the gel cutting. You can then perform gel excision safely. The Service Bio SCG W6000 Multifunctional Fluorescence Imaging System is an all-in-one instrument that integrates fluorescence Western blot imaging, Western blot chemiluminescence imaging, tissue fluorescence imaging, fluorescent protein expression in cells or microorganisms, protein gel imaging, as well as nucleic acid gel imaging and gel excision.